Today we're going to tackle Chapter 17 of Campbell Biology, titled From Gene to Protein, which is part of Unit 6 in AP Biology. Hey guys, this is Mikey from Avo Prep Academy, and on this channel we cover AP Biology content, and today we're moving on to one of the most important and challenging section of AP Bio, which is gene expression. Oftentimes we refer to our current model of gene expression as the central dogma of biology. The model on the screen shows this directional flow of information from DNA to RNA and then to eventually protein. The process of copying that genetic information from DNA to RNA is called transcription, and the processing of that information from an RNA molecule into a three-dimensional protein is called translation. In today's video, we'll discuss these ideas in three parts. 1. The idea of information and how the genetic code can manifest as three-dimensional molecules that we call proteins. 2. The specifics of the transcription process and the RNA processing that follows the synthesis of RNA in eukaryotic species. And 3. The process of translation, including that bit on protein targeting at the very end. So let's begin with part 1. First, I'd like to show you a picture. At first, it's relatively unassuming, with the tobacco plant on the left glowing in the dark due to a firefly gene that's been inserted into its genome, and the pig on the right with its glow-in-the-dark nose from a jellyfish gene. While this picture can seem fairly inconsequential, it actually hints at a super important theme in AP Biology, and that is the universality of the genetic code. The information found in our DNA and how it's transcribed and eventually translated into proteins appears to operate in the exact same way in every single organism. So in this picture, a gene from a firefly has no problem producing the right gene products even when found in a plant nucleus. And the same is true for the pig and the jellyfish gene. Imagine an app that you download on your iMac being compatible with a MacBook. It works on both devices because the programming language and the operating system on which it's running share the same code and origins. This is one of those major pieces of evidence that point at a common ancestry, indicating that our last universal common ancestor passed on this mechanism of gene expression in all the descendant species, including us. So the question is, what is this code and how does it store genetic information? So we begin exploring this question by revisiting the structure of DNA. Now you may recall that DNA is a nucleic acid with two anti-parallel strands of the nucleic acid and those nucleotides acting as monomers. Within these nucleotides, the most important bit is of course the nitrogenous bases. The four bases of our DNA, adenine, thymine, guanine, and cytosine, seemingly form sequences as they exist along this long polymer. Of course, the Chargaff rule applies as A's and T's and G's and C's being matched on either side of the DNA molecule appear in that double-stranded DNA. What we learned in the previous chapters, though, is that our chromosome is little more than a super long strand of DNA with some proteins thrown in for structure. Focusing solely on the DNA, though, we can start to see that some segment of that long DNA molecule can contribute to holding a discrete set of data that we often refer to as genes. Of course, in this chapter, we can engage with that idea of the gene with a slightly greater resolution, seeing them in codes of A, T, G's, and C's instead. For that added bit of detail, an average gene may contain some 10 or 15,000 base pairs in total, including both the coding and the non-coding regions. And one more important bit of information to add here is that the genetic information in its sensical configuration is always encoded on that DNA molecule from a 5' to 3' direction, reiterating the importance of directionality in nucleic acid structures. Now that we know how genes are sort of structured, let's talk about that information. In an earlier video, I mentioned that information can be encoded if we had codes, a sequence of those codes, and the general consensus of the direction in which we read and understand those codes. The example that I used was the word dog, where the alphabet letters that acted as codes would be sequenced with D, then O, and G, and when reading from left to right as we conventionally do, gave us enough information to know that we're talking about a canine animal. The same thing is true with DNA. With the evolutionary consensus of encoding genetic data from a 5' to 3' direction as the basis, we now see that the sequence of those four nitrogenous bases into some thousands of base pairs could encode information, as it were, within a segment of the larger DNA molecule. But the question is, what is this information for? And looking back to our diagram of the central dogma, it seems clear that the information in our genes must be used to produce proteins. So let's just take this brief opportunity to do a quick review of proteins. Proteins are created from one or more polypeptides, but for the sake of this example, let's just say that some protein is made from the folding of a single single polypeptide. So what is a polypeptide? Well, it's a polymer of amino acids, which incidentally were sequenced together from the N-terminus to the C-terminus with those some 20 possible amino acids strung together in a particular order. Now, this order, of course, resulted in the chemical interactions between those amino acids that naturally folded this polypeptide into a three-dimensional protein with its function arising from its shape. So it seems like there are some similarities between DNA and proteins. One, they're both linear molecules in that DNA has 5 to 3 prime, while proteins have an N-terminus to C-terminus. Two, they both contain monomers, and while DNA uses the 4 bases, proteins have those 20 amino acids. These similarities cannot be emphasized enough because it turns out that the information found within the sequence of a gene is in fact correspondent to forming the sequences found in our polypeptide chain of amino acids. So here is a simple illustration of how all this works from start to finish. But more importantly, here is that dictionary, so to say, of how the sequence found in nucleic acids can correspond to amino acids that ultimately formed that chain of polypeptides. Now, it is worth noting that in this table, we do see the replacement of thymine with uracil as the actual nucleic acid used during the translation process of nucleic acid data to protein data is in fact RNA. But that notwithstanding, it is clear that 3 nitrogenous bases can be used to denote an amino acid, which, going back to the picture shown earlier, can give you a chain of amino acids by using a slightly longer chain of nitrogenous bases along the nucleic acid molecule. So here is where we're introduced to the phrase of the triplet codon, with each codon representing a combination of 3 nitrogenous bases that correspond to an amino acid. What's important to keep in mind though is that combining 4 nitrogenous bases into a triplet would actually produce 64 possible combinations where we only have 20 amino acids to encode for. As such, we see what we call redundancies within the genetic code, referring to multiple codons that can actually designate a single amino acid. And we also see 3 codons, UAA, UAG, and UGA, reserved for what we call stop codons, which can act like a period at the end of the sentence in terms of data transmission. So at the end of the day, if we can take a sequence of bases and translate them into a sequence of amino acids, and if the resulting polypeptide or protein can fold into a shape corresponding to its function due to the sequence of those amino acids, then we've been able to successfully transform chemical data on a DNA molecule into a real 3-dimensional workhorses of the cell that we call proteins. Moving on to part 2, let's take a look at the process of transcription in greater detail. Transcription refers to that first arrow that extends from DNA to RNA. The RNA that we produce at the end of this process is referred to as mRNA, with the M standing for messenger. For now, let's not worry too much about why certain genes are expressed in which cells at what point of an organism's life, but focus solely on simply the mechanisms of transcription itself. Transcription is divided into 3 stages: initiation, elongation, and termination. Let's tackle initiation first. In order to understand initiation, let's have a better look at what a gene actually looks like from a slightly broader perspective. What we see in this image is the DNA molecule with its 5' to 3' and 3' to 5' with a large segment in the middle called the transcription unit. This is the part of the DNA and its sequences that will be copied onto a messenger RNA molecule. What's more important for initiation though is the segment labeled the promoter. This is sort of the start of the runway if we were to imagine a plane taking off, where the initiation and the preparation for transcription process will take place. The promoter is still sequences of DNA, and a eukaryotic promoter specifically commonly contains a region within called the Tata box, named for its TAA sequence within. To begin the transcription process, we first have the binding of very important regulatory proteins called transcription factors. These transcription factors are yet other proteins that have the capacity to recognize the promoter and begin binding to it. Binding of the transcription factors initiate then more factors into joining, and even some non-protein substrates like vitamin D to help get the process underway. Finally, an RNA polymerase binds. This is a large complex protein that carries the responsibility of reading the DNA molecule and synthesizing an RNA molecule as a copy. With the polymerase attached at the promoter, we now have what we call the transcription initiation translation complex. Now we're ready for elongation. In this part, keep in mind that any new nucleic acid must be synthesized in that 5 to 3' direction. This means that if the genetic information is stored from a 5 to 3' direction, then its complementary strand that we call the template strand can be read from the 3 to 5' direction while forming an anti-parallel RNA molecule, which then is synthesized in that 5 to 3' direction. And I know that this can all get pretty confusing quickly, so it kind of helps to draw all of this out on paper, especially if you need that extra clarification with these primes and directionalities. But as the polymerase unwinds a small segment of the gene, about 10 to 20 bases at a time, the template strand allows for the natural matching of the ribonucleoside triphosphates that enter the polymerase to form a corresponding hydrogen bonding with the correct complementary base. So for instance, an exposed thymine on this template strand would allow for an adenine containing ribonucleoside to bind and chemically confirm that the right match has been made. This allows for the continuation of the process, correctly matching the template bases to the incoming bases as the polymerase progresses forwards. Of course, it would rewind that DNA behind it as it continues to move. By the way, this happens at a rate of about 40 nucleotides per second. In termination, the polymerase reaches the end of the transcription unit. This is typically detected by the polymerase through the recognition of certain sequences in eukaryotic cells that begins the dissociation process of all parties involved. This would also free that RNA molecule from the polymerase. And it's at this point that I like to differentiate what's happening in prokaryotic versus eukaryotic cells. As you know, prokaryotes don't have a nucleus, so the whole process just sort of happens in the nucleoid region of the cytoplasm. However, with eukaryotic cells having a nuclei, the process of transcription would happen within the nucleus, separate from translation. This means that the RNA molecule produced from a eukaryotic RNA polymerase must be transported from the nucleus into the cytoplasm for the remainder of the translation process. But there's one more thing that the eukaryotic organisms have to do before this export even happens. And here we get to learn about something called RNA processing. So RNA processing happens in eukaryotic organisms, and it involves three major alterations to that RNA molecule that we'll refer to as the pre-mRNA. First is something called 5' capping. Here we see that a modified guanine nucleotide is added to that 5' end of the pre-mRNA molecule, forming a cap that helps with the export process of mRNA from the nucleus to the cytoplasm, helps to protect the mRNA from degradation by hydrolytic enzymes there, and lastly helps to bind ribosomes, which ultimately uses the mRNA in its process of translation. Second is the addition of a ton of adenines at the 3' end of an RNA molecule. This is called polyadenylation and is typically performed with the recognition of the polyadenylation signal AAUAAA. Typically, some 50 to 250 adenines are added, which then further protects that mRNA from degradation in the cytoplasm. Last but not least, we have RNA splicing. This one requires a little bit more explanation. Looking at this illustration, we see that the transcribed RNA has regions denoted as exons and introns. Interestingly, what eukaryotic cells do is to remove these introns and splice together the exons in creating the final sequence of bases that will be used in the translational process. So a couple of questions arise here. Why cut out introns while keeping in exons? And as silly as it seems, much of the vocabulary here is from English and not Latin. So exon comes from the word expressed, while intron comes from the word intragenic. It's pretty annoying, but it is what it is. But the bigger question is why? Because it seems that prokaryotes don't need to do this extra processing, making the process a little bit more efficient, right? Well, the truth is a little bit more complicated. It appears that a eukaryotic proteome, or all the collection of all the proteins found in a cell, is always more vast than its genome, or the collection of all the genes within an organism. This means that there are way more proteins than the number of genes could account for. This is because of something called alternative splicing. In alternative splicing, we see different exons being kept in the final mRNA depending on the variant of the protein that we want to produce. So for instance, you may want to keep exons 1, 2, and 3, or 1, 2, but 4, ultimately resulting in multiple permutations of exons that would result in varied proteins from a single gene. This is sort of like data compression, where you can get more data out of a smaller set. And if the decoding rules can be understood by the splicing mechanism, then we can carry way more data within a smaller genome. genome. This would ultimately result in faster replication, which, as you can imagine, would provide a selective advantage in the arena of natural selection. Now that we have the full-fledged mRNA in our eukaryotic cell, let's get that molecule into the cytoplasm and actually translate that information into a working protein. So here is part 3 on translation. So the process can be pretty complicated, but let's first tackle the problem of this word translation. Taking DNA or RNA information and reading our codon charts to figure out the amino acid sequence is easy for us because we have brains, but cells don't have brains, and somehow this transformation of data in its nucleic acid sequence to an amino acid sequence happens naturally. So let's take a look at an analogy of how this might work. I'm sure many of you guys are taking a second language at school, be it French, Spanish, or Latin. There may come a time where you need to look up a word that you don't know. So let's imagine for a moment that you don't know the word gato in Spanish. Now, of course, you can ask Siri or Alexa.
Parece que estás buscando la palabra gato.
But before all of that, we used to use a dictionary. In the dictionary, what you'll find is the word gato, perhaps noun, and its English meaning, which is cat. But notice how in order for you to translate that information, you need to have something that contains both Spanish and English simultaneously. And in our cells, transfer RNAs, known as tRNAs, do just that. So let's take a look at a tRNA molecule. Here we see that tRNAs are in fact RNA molecules with A's, U's, C's, and G's just folded into a shape that in this diagram appears to be a T. But what's important is the directionality of how we typically present tRNAs. It goes from 3 to 5' with this bottom loop that we call an anticodon. And on that 3' end, well, we have an amino acid attachment site. So what's cool is that in this picture, a 3 to 5' of AAG being our anticodon would correspond to a 5 to 3' mRNA codon that reads UUC because of complementary base pairing. And on this very specific tRNA, UUC's corresponding phenylalanine could be found on the 3' amino acid attachment site. So tRNA molecules act as this very dictionary page that contains both the nucleic acid data and amino acid data. And just keep in mind that we refer to tRNAs with amino acids attached as aminoacyl tRNAs, which are then synthesized by an enzyme called aminoacyl tRNA synthetase that matches the right tRNA to the right amino acid. And we should expect to see many different tRNAs to correspond to the 20 amino acids and multiple codons that they must recognize. In fact, we see about 45 tRNA molecules in organisms, far fewer than the 61 possible codons, but that's a topic for a different video. Now that we have the tRNA, let's take a look at the machine that can create the polypeptide from a messenger RNA, the ribosome. Now, ribosomes come in 2 parts, the large subunit and the small subunit. They're also associated with ribosomal RNAs that assist in the translational process, but are mostly made from proteins. Initially, these subunits exist separately in the cytoplasm. So now to the mechanisms of translation, we also see those 3 stages: initiation, elongation, and termination. So be careful here not to confuse them with the same names used in the transcription processes. But let's begin with initiation. The messenger RNA, having been processed in the nucleus in eukaryotic cells, exits through the nuclear pore and enters the cytoplasm. Here, the 5' cap assists in connecting that mRNA with the small subunit of the ribosome first. The small subunit attaches towards the 5' end and begins to scan down the length of the mRNA looking for something. That something is the start codon AUG, which corresponds to methionine. Once AUG has been found, the next thing that happens is the binding of the tRNA that has an anticodon of UAC, which incidentally forms a complementary bonds with AUG, and it carries that first amino acid of the polypeptide, methionine. Once this initiator tRNA binds, the large subunit attaches, forming a full ribosome. But what's important to note here is that the large subunit has 3 sites or regions within: the exit site, the peptidyl site, and the aminoacyl site. During initiation, the large subunit comes over top of the initiator tRNA, placing it in the P site. Now we're ready for elongation. You see, the aminoacyl site allows the next tRNA to come into the ribosome. If the correct anticodon matches the next codon, then this match facilitates the formation of a new peptide bond between the amino acid of the tRNA in the P site with that of the A site. Here is where things can get a little bit tricky though. At this point, the small subunit moves 3 bases forward, placing the tRNA in the A site into the P site, and the tRNA from the P site into the E site. The tRNA, no longer attached to its amino acid, is then released from that E site just to be recharged by the aminoacyl tRNA synthetase, while the A site is now open again for the next amino acyl tRNA to come in. This process will repeat, continuing to move down the length of the mRNA, placing each and every amino acid in a sequence that corresponds to the base sequence. This process requires energy and it typically comes from GTP. Now, termination is a little bit more specific in translation as opposed to transcription. Earlier in our codon chart, we saw UAA, UGA, and UAG as being the 3 stop codons. When the elongation eventually reaches to place one of these 3 stop codons in an open A site, the molecule called the release factor comes into the A site instead of an tRNA molecule. The release factor will then release the ribosomal subunits, cleave the bond that holds the very last amino acid to a tRNA, and this in turn should make everything return to normal. The 2 ribosomal subunits are separated, the mRNA in the cytoplasm is still just hanging out, except now we have the polypeptide, which probably has already folded into the structure that it needs to be. While that primarily covers the process of translation, there's this one extra bit of information that the chapter presents: protein targeting. Back in chapter 6, in discussing the endomembrane system, we had discussed the idea of protein export by the use of the endoplasmic reticulum, the Golgi body, and the vesicles that eventually fused with the plasma membrane to perform exocytosis. Here, we're just given a slightly better picture of how that protein enters the ER lumen. So yes, all the translation process begins in the cytoplasm, but a short sequence of amino acids called a signal peptide will engage with a signal recognition particle, which then carries that mRNA and binds to a channel on the endoplasmic reticulum that opens a channel, which then allows the protein synthesis to occur into the endoplasmic reticulum. So whether you're making proteins to export or to place within a membrane-bound organelle, or even create channels or other intermembrane proteins, the ER is the place that ribosomes can deposit its polypeptides. And this is also why the rough ER appears the way it does, due to all of these ribosomes that are attached to the ER at any given point in time. Okay, so that about covers the bulk of this chapter 17 on gene expression. The only thing left to cover in this chapter is mutations, but seeing as how mutations are relatively self-explanatory after having discussed what we did, I'll keep the topic out of this particular video. If you'd like to see a whole other video on mutations, let me know and I'll make that happen for you guys. Well, thanks for staying to the end of the video. If you found this video useful, consider clicking that like button and subscribing to the channel. And as always, this has been Mikey with Avo Prep Academy. Have a great day.